am elated to welcome Ramanujan, the founder and promoter of Propinquity Genomics and Sciences. He is also the co-founder and chief scientific officer and chief growth officer of Oncophenomics and a board member SAB and chief advisor Genomics Global Business. So, Ram is a versatile personality with 32 years of functional biology experience, and he has worked on microbiology, genetics, enzymology, protein chemistry, microbial physiology, nucleic acid enzymology, genomics, and IT. He has work experience from global leaders spanning 4 countries. As a committed individual with perfect combination of biology and IT, he has worked in various types of organizations like product and service sectors and is blessed with the skilled eye for the detail and exceptional in timely execution of projects and succinct performance. He is the proud owner of Make in India New to Earth molecules, particularly on his discovery TJs from B subtilis 168 for various application bits competitions, hands down like that of Ajinomoto of Japan. He is the only one in Asia to have developed novel neurotrophic factors and neurotrophins as rhDNA derived therapeutic protein replacement therapy, which he conducted many studies in USA Pharma during early mid-90s. New biologics is core focus area in India, and he is motivated to elevate Indian science to global forums. Ram's core competent areas in research and development include large molecule drug process development, downstream processing, replacement large molecule therapy, novel biologics, monoclonal antibodies, biosimilars and biogenerics, genomics, genome informatics, bio-IT, bioinformatics, fractal biology, fractal genomics, oncoabs, genome-guided therapy, fractal methods-based diagnostics, and cancer therapy.
A very good evening to everybody and welcome to yet another episode of Eclectica, the Different News. As you can see, I already have my fascinating guest today with me on this platform. So I welcome you, sir. I think it's an honor to have you on this show, and it is fantastic that you accepted my invitation. I'm very obliged, and thank you so much for being on my show.
It's a pleasure.
So, and as I know that your journey as in genetics or genomics, however we put through, I think it started long back, and your story is very, very inspiring. So instead of me telling about it, I think it would be very interesting to know how your journey almost started at your teens back in maybe, I don't know, it might be 1980s or before. So I think it will be very interesting to know and understand from your journey. If you can really share, it will inspire all of us.
First, I would like to credit my teachers, without whom I would have not been here. First, I would like to take my botany teacher, Professor James Clement, when I was 15 years old. He handed me the genes book from Benjamin Lewin. That was the start in the summer of 1980. I started reading the genes book. I was very inspired by the nature of the biology. So I studied chemistry and biology in my +1 and +2. In 82, I joined my biochemistry, which was a university department in PhD College of Arts and Science in Coimbatore. It was started in 70s. And that was the only college which is offering the biochemistry along with Avinashalingam College in Coimbatore, which was a sleepy town at that time. So it goes from 82 to 92. It was all research and training. And in 92, I entered the biopharmaceutical industry. So my education was done in the United States, and my training was also in the biopharmaceutical company, startup company at that time, which is a very big company right now. It is called Regeneron Pharmaceuticals. And I went to school, New Mexico State University in near El Paso, Texas, and University of Kansas, Lawrence, Kansas. So I was studying for about 4.5 years, and then I went to industry. From there on, in '95, I happened to come to India because of family reasons. And in
Mhm.
'94 November, when I was visiting India, I met the Biocon group. At that time, it was called Biocon Unilever Group. And then in
Right.
'95 December, I joined them. Although my experience was in novel biologics,
Mhm.
biopharmaceuticals for neurobiology diseases like ALS, amyotrophic lateral sclerosis, I was working on recombinant human CNTF for the second phase and third phase clinical trial in the US. I was part of the process development group there, headed by some eminent people. And the research division was headed by Dr. George Yankopoulos, who is a biotech billionaire researcher. And those research that they were doing, which was converted into products, they were been
Right.
going through the clinical trials in the US. So to continue a short brief, to continue this journey, in '92, when I joined Regeneron, it was a very small company. Now it is more than 10,000 people.
Wow.
So, and that is the type of company I worked for in the initial stages when the spin out was happening from Columbia University. So that is when I got my biopharmaceutical novel biologics, discovery biology experience. Before that, I was a discovery researcher, but in RNA sciences. So then
Hmm.
I picked up the RNA sciences in University of Kansas, and then I went to Germany and Japan. I worked on RNA sciences, ribozymes. Basically, these are catalytic RNA and hammerhead ribozyme and group 1 intron, and these are different ribozymes that were discovered by Tom Chuck and Sidney Altman, Nobel laureates in 1980s. And my boss came from that lab, and in fact, he is a senior to Jennifer Doudna, whose his name is Dr. Peter Geigenheimer. I should mention one more Dr. James Hageman, who was my boss in New Mexico State, is a PhD from Daniel Atkinson Lab and Paul D. Boyer Lab. And Paul D. Boyer is the ATPA's Nobel laureate in the
Yeah.
70s and 90s actually. So I had a privilege of working with some top leaders in the field of ribozymes, RNA biology, and as well as in protein chemistry and biologicals. I brought that to India. I asked a lot of people here in India, are you willing to do the biologicals? So at that
Mhm.
time, '95 was very early time for India, and it was not
Yeah.
known. Biologics were not known in India. So what happened? I pitched myself as a genomic scientist. That is when
Mhm.
I decided to get into informatics. Genomics is informatics. This was not realized in '95, '96. So we were very early people in that time. So I jumped from when I came from Germany. I met one Dr. V.N. Balaji, who came from Monsanto. He played a pivotal role in my career in bringing myself as a bioinformatician. Then I switched to a couple of companies like Satyam Computers and DSQ Biotech. I was in bioinformatics as a very early entrant. Then,
Hmm.
when the second generation genomics happened, I jumped into that. That was in 2006. And then I picked up the genomics, and I have been ever working with genomics. Another important crucial point happened in 2011. My introduction to Professor Peliones, Andras Peliones, he brought fractal genomics into play, the fractality of the genome. So for the past 13 years, I've been associated with him. He is 80 years old, and he is an Hungarian American biochemist and a genomic scientist. He's a triple doctorate. He is a doctorate in computer science,
Wow.
biophysics, genomics. Right. And he was a professor at New York University. So
Mhm.
he taught me this from the fundamentals of the fractal analysis, fractal genomics, which is what I'm going to talk to you today about,
Right.
which is what propagated genomics is doing right now. And what I did was I got introduced to French researchers, Russian researchers, then
Mhm.
Switzerland researchers, then Bella Balabas, mathematician in UK. So these people are top level people in this area. Fractals are very interesting area, biomathematics and computer science and IT. So many things are flowing in. So interdisciplinary in nature. So that is where we are approaching the genomics as informatics. That is exactly where I am right now, approaching the fractal genomics.
Amazing. Amazing. I think we like. we always talk about interdisciplinary, but then I think you are someone who is actually a living legend or a living example. I can put it forward, and the kind of variety in in your career till now you have done, this is amazing. I think this is itself a inspiration and encouragement for all of us, and I think sir, like the young people who are still confused where to go, I think when they listen from you and when they understand that this a single person can do so much, so I think they get a lot of inspiration. So I think, sir, it
Yeah,
is time
I
that
think the start early promise, as Jim Segaman wrote a book in ACS. The students in the United States start as early as 17 years in the lab,
today.
and
So,
our
sir, I think we are all waiting for the interesting part of this conversation, because what you said, fractal genomics, fractal, this is something. I mean, genomics quite has become a buzzword now, and particularly COVID has taught us. I think every layman at least knows what is RT-PCR, and so from that point, sir, if you can simplify for us, and if I can ask you a very straight question, that what is the fractal genome, and a tag question would be, and how is it different from the traditional view of the genome?
yeah. So let me point out to you, the genomics is becoming a household name, but not many people understand genomics.
Right.
And RT-PCR, anybody can in India say about RT-PCR, I have taken a test, but they don't really know how it is done. So these are 2
Exactly.
points. The third point is, your question is very valid. So the genome is a fractal. What is a fractal? Fractal is a self-similar repetition. The self-similar repetition is, for example, you see snowflakes. This was introduced by Mandelbrot in 1975. Benoit Mandelbrot. So the fractal term was coined by him in 1890s when mathematicians were there. They had known, but it was coined by Mandelbrot, Benoit Mandelbrot. So the point is, self-similar repetitions are nothing but, when you take a cauliflower, you see these self-similar repetitions. When you go to a mountain, okay, the cone structure, that is a self-similar repetition. And you don't go to anywhere, any other place, go to your Varadaraja temple. You will see a lot of gopras in Srirangam and other places. They are all temples of fractal structures. So I can give you a simple example. If nobody understands fractal, go and ask your forefathers in India. They will tell you about how they built the fractal geometry temples. Indians know this. And this was understood by Americans and Europeans much better, because they knew the mathematical prowess of India and later the computational prowess of India. That was the main reason Andros Paleonis was very interested in India, coming to India and propagating his discoveries. But unfortunately, our Indian companies are not ready for such thought provoking 10, 15 years ahead of his time technology. So people are, I will come to that, why I say that. The point, next point is, when I say statistical diagnosis, we use a statistical technique for the diagnostic value, probabilistic prognosis. So it is actually what kind of prognosis you are looking at. Third thing is precision medicine. There also we are looking at fractals only. So I will come to the explanation of those 3 points. Let me take you to the second point. Why is it fractal? Well, why would snowflakes be fractal? Why would mountains be fractal? So the question is, the genome is a fractal. Mr. President, the Eric Lander said in a Science magazine cover, genome is a fractal. So it is a known fractal globule structure which was depicted by Ares Lieberman in Houston and this Freudenberg in Harvard. They all did a technique called IC technology. They found the structure of the genome is as important as your genome sequence. So therefore, if you have any problem like a CNV, copy number variation, later it can be called as copy number alteration, you will still have the structure of the globule, fractal globule structure of the DNA altered. I will come to that point little bit later when I explain. Go ahead with your question.
I think that is very interesting, and the very visual, simple examples you gave, it is very easy to at least touch upon the fact that we get a visualization of what exactly fractal or fractal genome is all about. So, sir, how can a statistical diagnosis be used to identify genetic diseases as that matter?
Okay, so most of the mutations that you have seen, for example, is going to be SNPs or simple mutations, deletions, additions, or variants. We can call a variant. But this large C and V, copy number variants, they are called structural variants. In India today, they do so much of testing. We don't do structural variant testing actually. We don't know. We don't even
Yes.
have Indian population reference genome. We are depending upon a Caucasian genome. We have an Infinium chip, Illumina 4.0 is coming, which will have a little bit of Asian data, Indian data. But most of these companies which are working on are all working on Caucasian data. So if you want real Indian data, you have to reference genome. 4,000 ethnicities or different sects of people in India available. Therefore, when we need to sequence Tamil Nadu population, Karnataka population, Assam population, Manipur population. Therefore, we have to come up with a reference genome. Having said that, the point that I want to make today is our tests that are being done in India mostly are false positives or false negatives. The doctors will get confused about it because the
Right.
structural variants are not looked at. Okay, you look at some variants and say some doctor comes in and says, okay, I have a cancer patient, let us say cervical cancer or ovarian cancer. And the company that gives the data, it gives a false negative data. Then what happens? So therefore, validation is the key. You have to validate your data. If it is false positive or false negative, controls have to be in place. I have Indian population data. At the same time, I have Indian reference genome. This is the Asian data. I can represent it. I can look at the SNPs. I can look at whole genome sequencing. For cancer, most of the time, whole genome sequencing people are not doing. There is a TCGA atlas, but whole genome sequencing is not required. Therefore, people do exomics. Exomics is
Exomics.
very much done. So you understand what exomics is all about. So when
Yes.
they do whole clinical exome, today the market in India is a whole clinical exome market. I predicted that this market of whole clinical exome will be 10,000 samples per month. So 120,000 samples will be coming in. But the point is not that. The point is how effectively, accurately you can say these are not false negatives, these are not false positives. I represent these are actionable mutations. Based upon the actionable mutation, I can go for a precision medicine or the
Right.
therapeutic intervention that I can do. My
Hmm.
point is again to tell you that the fractal genomics here, what we are proposing, is a very advanced biomathematics and information technology associated. We predicted in 2012 that the pharma companies will be IT-led pharma companies. There won't be pharma companies anymore. There will be all informatics-led companies. And today that is exactly happening. It is genomics, that is informatics, and without IT you cannot do it. It is
Correct.
not possible to do human intervention all the time. We need sophisticated software. We need human intervention also. But the sophisticated software will help you to make that judgment by human intervention.
Right, sir. Right. I mean, there's a very new dimension of knowledge that is opening up by talking to you, and we really understand the real picture what is running. So coming to my next question, like what is probabilistic prognosis and how it can help in personalized
Predictive.
medicines?
When you do statistical, we have a counting method called box counting method. The box
Okay.
counting method is one of the techniques of the statistical diagnosis. That means we have to do statistical analysis. While
Okay.
fractal analysis, we do have a technique called box counting method. So we can actually get the fractal dimension. That is what we call it is highly mathematical. Okay, I don't want to go into a lot of details. There are several techniques of there. Second point is predictive in the sense probabilistic. Let us say
Hmm.
this is a topology that I am working on. So prognostic. So unless you know the diagnostic prognostic value, we cannot go for a precision medicine. Today, let us say we take thousands of drugs that are available. Thousands of drugs that are available for chemotherapy. Is the
Hmm.
doctor very clear which drug he wants to give? Or is it trial and error? Or if it is a
Trial.
surgical oncologist, if he doesn't have the genomic test, is he doing justice to the patient by doing a surgical oncology? Or is the tumor mutation board, that is TMB, what they call
Hmm.
mutation board, Does it have a genomic scientist along with other scientists like oncologist and a doctor and all of them? So what happens? Genomics is a genome guided therapy. In Mayo Clinic, there was a clinical trial, Beauty, breast cancer clinical trial. There, the first time in 2011, they did genome guided therapy. So we are far behind in this curve. So we have to catch up. So what I'm saying is that if Mayo Clinic can do it in 2011, this Beauty test, which is the breast cancer genome guided therapy, we knew this. So that is why we were proposing the precision medicine that we are proposing. I will come to this point, how I am proposing. See, what happens in the genome? There are fractal defects. Fractal defects are either copy number variations or change later to copy number alterations. So these genome defects, we are neutralizing. What happens? 2 meters of the DNA is stuck in a 6-micron DNA nuclear structure. How is it possible? It is a folded structure. It is a compact structure. So therefore, if you have these CNVs, it will clog the transparency of the 2D Hilbert curve. The 2D Hilbert curve governs the global fractal globule DNA. That is why Hilbert is a very old mathematician. I told you the Benoit Mandelbrot is 1975 phenomenon. So apart from Benoit Mandelbrot, several other scientists have been working on fractals. Okay. The question is that, okay, the transparency of the genome is clogged. And for your viewers, I would be more than happy to share one article which you can attach along with this interview, where it is clearly saying if you transparency of the DNA, if it is clogged, it will cause cancer. The genome, see, because the genome structure is altered. Therefore, the point that we are making is that the 2D Hilbert curve has to be clearly understood, and there is an equation. In this equation, Z is equal to Z squared plus C, where C is a constant. The square that I am talking about, it is a syntax that can vary. That will create a fractal defect. So harmless defect or a harmful defect, that is a question. So these are the called what you call as L-string algorithm. Sorry, L-string Lindenmayer systems. And these Lindenmayer systems are very much known to mathematicians. So here you see, this is not just genomics. We are working with mathematics, IT, computer science, and biology. It's totally interdisciplinary. You need to know a lot of things. Even biology also, basic biology like RNA systems, all these all this to be mastered. The one point I want to add here, what we were saying is, he proposed that Andres Milionis has proposed, PRGF. What is the principle of recursive genome function, PRGF? What it means is, DNA to RNA to protein. The original double axiom is Watson and Crick axiom, DNA to RNA to protein. But nobody knows it can recurse. So it is not a one way. That the protein we already know are binding to the DNA as a transcription factors.
Absolutely.
And RNA can recurse to DNA also, which we know from reverse transcriptase.
Correct.
Which was discovered by Baltimore and Howard Temin. So it never recurses it is the paradox. And if you understand Susumu Ono's junk DNA, 98% of the junk DNA and 80% is functional. That itself was not understood. Therefore, now we are finding, long non-coding RNA. We are finding many other RNA in a non-coding regions. So the non-coding region is equally important. Therefore, the coding region is important. Non-coding region also are playing equally. So these are genome regulation diseases. So the non-coding RNA is very important. Okay. Therefore, the ENCODE project, when it was completed, Francis Collins said, scientists have to think about long-held beliefs. You know why he said that? Because at that time, nobody believed in non-coding junk DNA. They believed in junk DNA, junk is junk, anything but. But now it is proven, junk DNA is not junk, it is not anything but junk DNA is 80% is functional.
Correct.
There you go. So now the point that I would try to make to you is, Please understand, basic biology has to be understood, and it has to be mathematics and computer science has to be applied. Then
Correct.
you will be able to analyze data in an analytical fashion. So we are actually proposing an IT-driven pharma, a hospital system in the ground floor where we will do sequencing, a analytics lab where we will do analysis, and clinical trial where we can establish the validity of the particular system. So in this regard, I would like to point out only one point, that we call this as an RNA system. We have two concepts. One is called covariant, contravariant functors. I don't want to go into too much detail about the mathematical terms here, but what it means, it is an RNA system, it is a fractal system, it is fractal gene. That is what Dr. Perianis named it. So at this point, I want to mention to you, I have Professor Jean-Claude Perez in France. He is a collaborator with Luc Montagnier. Professor Luc Montagnier is the first person to discover HIV strain.
Yes.
And Luc Montagnier and Dr. Jean-Claude Perez published that the COVID genome has got fractal aberrations. Do you know why? The problem is in COVID, the COVID genome, which is the RNA genome, they found exogenous sequences. They found exogenous sequences. So the lab leak theory may be true. I don't know. Now FBI says and the government says that is a lab leak theory has been validated. But Professor Pelionis and Professor Jean-Claude Perez and Luc Montagnier proved that these exogenous I have the paper. Exogenous sequences are there, and that is probably the reason these guys are actually responding to ivermectin. Why would it respond to ivermectin? That is the question I have. Second question I have is, this genome, RNA genome, now you say, has got fractal aberrations. So it is very clearly shown. I even put it on my LinkedIn page. So that's very important. How to neutralize that? That is the question. So when you come to the point of neutralizing the fractal defect, when it comes to the critical point, how to neutralize the fractal defect, now that is where the precision medicine comes into play. No, you can neutralize the fractal defects with the precision medicine.
Okay.
It is possible. So we know that we have to do the clinical trial, we have to do all the things, I told you, IT-led pharma, clinical trial, sequencing, analytics, mathematics, software-enabled approach, algorithm should be with us. That is where Pelionis filed a US patent, fractal organisms, fractal organs, fractal organelles. He filed the patent. So fractal system, how the fractal system works. I'll be happy to share that patent as well. So we're looking at Indian companies. Can anybody would be interested in this genome informatics? Would anybody would be interested in the software-enabled approach? You call yourself as a software giant, the power, but we have not gotten to the nitty-gritties of the genome informatics. So it is still hanging around, So that is where my entire 12, 13 years I have been spending trying to find out a good match in India as a BRICS country to take part.
Amazing, sir. And what a clarity of such a intricate, complex science. Because many of the things that you are telling, basic biology, yes, I am aware of it. But the way you have, put it up like a platter, I think at least through this conversation, relevant people, you have created that path of thought provoke. I mean, you have provoked them to think. I think that is a very good thing.
You know very well we are ahead of time. This is one of the problems. And Professor Pelionis is very ahead of time. He is 81 years, 80 to 81 years. And Jean-Claude Perez is another 80 plus years. And Luc Montagnier is no more. And I have another collaborator, Professor Sergei Petekov. He is not my collaborator, he is a mentor for me. And he is in Russia, laureate of science. And he is very close to the government there in Kremlin. So they all are, I am blessed to have such people. And also, we know Gabriele Losa in Switzerland. He's a pioneer in this actually. And another person is Professor Bela Balabas. He's a mathematician. In fact, he has praised Srinivasa Ramanujan, my own name, as a very big mathematician of this century. And in fact, he himself is a big mathematician. This Bella Balabas. He is from University of Tennessee and UK. So these are the people, and I have a couple of other good PhD postdoctoral, all foreign trained people as the International Fractal Alliance that I convened, and I'm trying to do my best, but finding funding for this kind of novel approaches are not very easy because
Yes.
you need the people who are funders look at equitable returns, and here it involves a lot of work. And maybe the only way to go about it is to find money in the US.
Right, sir. Right. I'm sure this approach, the kind of challenges you have taken up, and particularly bringing all this to India and trying to build it up here. I mean, your honest interventions and your honest vision, I think, is just matter of time. I mean, all the enlightened mind that you have grouped together. This is itself a very big thing.
Yeah,
I must, humbly put it
I think
to
so.
you.
They have been very kind to me, and they
Exactly.
knew that I'm trying to bring something to India,
Hmm.
and they have been supporting me. And this is not ordinary support; it is 13 years of support.
Correct. Correct. So, sir, taking our conversation further a little bit deep towards the deeper end. So, how can cancer precision therapy be tailored to an individual's genetic makeup?
Okay, so now there's a lot of tests are coming in. Like for example, you might have seen in the press, we are whole genome sequencing we are doing, and we
Yes.
can tell you predisposition and
Correct.
Infinium 500,000 markers that I'm doing. Industry Infinium 4.0 is coming. Some Asian data is there. I would do the SNPs. Now, what all
Yes.
this tells, it doesn't. The problem is it is predisposed. So there is not a precision medicine. The precision medicine is about actionable mutations. There, I already told you the CNV nobody does in India. All
Correct.
changed the name CNA. So they are looking at point mutations. Like for most of the tests in India were doing previously 5 years back, 7 years back, all like BRCA test, Oncotype DX, or
Correct.
Mammoprint, this kind of single gene tests. So now
Hmm.
that the whole clinical exome has come, now we can do the whole clinical exome, 22,000, 23,000 genes. Okay, we are
Hmm.
doing. Now, actionable mutations, the variant calling, there are several good programs are there, namely PRI and ARVAS, and the variant calling programs are there. But even then, I heard they are very expensive for India. And many people don't go for it. They put up a factory of people, 30, 40 people, and do human intervention. So the
Hmm.
question that comes to my mind is how exactly they are validating this actionable mutation to give the doctor that pleasure that yes, I have the validated actionable mutation. If
Correct.
I take this, I go with this. I go with this particular monoclonal antibody, or I go with this
Hmm.
small molecule as a chemotherapy. How do you tailor make that? So the point is it genome guided? That is where the crux. If it is
Hmm.
genome guided, if your data and analytics are reasonably very good, you can make some clear judgments. But if you are
Right.
giving false positives and false negatives, that doesn't serve the doctor, and over the period you will lose his credibility. So once the credibility lost with the doctor will not come back. So it is very important for the genomics industry to establish with the doctors. And that is one of the reasons this is underpenetrated industry,
Yes.
actually.
Yes.
Why is it
True.
underpenetrated? It is like only uh, 193 crore or something like that I heard, which I have to confirm in
Hmm.
21-22 is the clinical genomics. What is India's population? 1.5 billion. Where is the vast resources are being wasted here? Because this is a very
Correct.
small market for pharma market is 40 billion. So the biotech
Correct.
market we call it as a bioeconomy in hundreds of billions of dollars. But when it
Hmm.
comes to specific genomic tests like clinical
Hmm.
genomics, can anybody give me a correct number? Today nobody can give me a correct number. They are all approximations. But I don't think the market is big enough. It may become big. I'm not saying it will not begin up because sub $100 genomes can happen. Ultima
Yeah.
is coming up with $100 genome. Illumina is already $200 genome. And
Yes.
you're talking about elements biosciences, which can come up with equally. But the problem is a giant like Illumina is still unbeatable giant, very big
Hmm.
company. So their
Absolutely.
resources. So look at some newer comers, they will take a little bit of time. So can we achieve $110 genome? That will be very far away. a little bit far away. So can we achieve $100 genome? That we need to try it out because we don't have those Ultima machines in India. The only machines that we have is Illumina X and X Plus. The X
Hmm.
Plus we calculated it is working out to $60 for 30 GB, 10x low coverage. So if you are looking at that,
Hmm.
it will work out to $200. Still, it is $200, 16,000. So if you offer a test at 12,000, you will still not make a profit. So you have to give it free. So we
Hmm.
are responsible for the patient-centric approach. This is not actually a profit-centric approach. So that is
Hmm.
what we are saying. I'm actually with a company called Genomics. I'm on the
Okay.
scientific advisory board, and I'm
Okay.
also on the scientific advisory board of the core diagnostics, and they call it as
Oh,
core genomics.
yeah.
Actually, we are trying to do this patient-centric approach, and the
Ex-
promoters are in agreement with me that we should be patient-centric, not the profit-centric. So we need
ab-
like-minded people to go to the doctor and say, "Sir, we don't want to charge you so much. We want to be at the modest level, but we want to be
hmm.
patient-centric. We will work with you for some time. We will validate this data, and then I will give you confirm to you that your credibility is established." So the point
Exactly.
is, this is not some telecom business, to go about 400 million people. That is not the market. I
Hmm.
made my point.
Yes, absolutely, absolutely. So, sir, like we are talking about data and data collecting and genome sequencing, all the data. So, everywhere in these process, I think ethics or ethical considerations come into play. So, my next question would be directed that way. So, what ethical
I, I draw
consideration?
your I draw your attention to Dr. Vinod Skaria. He has
Yes,
been in touch with me. He has been
yes,
putting a lot of information about people sending data outside of the country. It is
correct,
frequently happening. Nobody is exception to the rule. It is happening. But the question is, can Indian data be allowed to send outside for sequencing? Dr. Vinod Skaria and myself, we are subscribing to the fact it should not be sent
yes.
at any cost
Hmm.
because you are compromising Indian security. This is not just Indian security. I will come to one point. The CIA has published a paper that
Hmm.
CRISPR is a bioweapon. Anybody who has got access to the CRISPR can be a bioweapon. Now, China. So the point
Yeah.
is that I think that security should not be compromised and privacy. See, for example, I give you my data. Actually, I have you have to get an informed consent whether I should
Absolutely.
share my data or not. This
Hmm.
informed consent, if I say no, you erase it from your database. That's it.
Hmm.
Be it big company in US or big company in India, they have to erase it from their repository.
Correct.
That is the
Correct,
point
sir.
of consent. That is the privacy, ethics, law of the land. So these are all the
Right.
things that we are not planning to do because see, at the end of the day, profitability should not be compromised for the national security.
Absolutely. Agree. Absolutely, sir. Absolutely.
So
Sir,
today is okay. You give it to some Indian pharma companies your genomic data. What happens if it gets into Merck and Pfizer? That's Indian
yeah,
data. There is
correct.
already Dr. Vinod Skaria has mentioned that
Yes.
this data has gone out of the country.
Yeah, I think you shared one article and I went through it and it was about he also filed an RTI and he's trying to figure out and that's a very noble.
He is very vocal about it, and we are all vocal about it in the LinkedIn.
Correct, correct, sir, correct. So I think this is something which is of real concern, and we need to be aware also about it. So people are not aware also, I think. So
No, there is a
yeah,
problem. The awareness is very limited actually because they think, see, genomic national security issues is as equivalent as biological issues.
correct, correct, absolutely. Anything, I mean, anything can be figured out of this kind of a data, as you mentioned already. Yeah. So, sir,
Today,
what do you
in
think?
the dawn of the gene synthesis machines like Codex 3250, anybody
Right.
can synthesize any gene, oligo, anything. So I don't know. I don't know.
Yeah. I, let us hope for the positive aspect of science and let us hope for the best. I
No,
think
it's
that is
only
that
word
is
of caution. I'm not saying it is happening.
Yeah.
But even people like us who got our machine in 3250, we have to
Huh.
comply to the rules and
Correct.
regulations. We are
Correct,
not
sir.
doing any genetically engineered organism. If we want genetically engineered organism, we have to go to GEAC or RCGM. We have to get our permissions.
Absolutely.
So my point again, I'm coming to the point that this industry is unregulated. This genomic testing first
Hmm.
regulate it.
Hmm.
So my humble submissions to the regulators, unless you regulate this industry, this is going to go average.
Yeah. Very true. Very true. So, sir, what do you think, like how the post-genome era changed the way we approach, medical research, or a lot of buzzes there regarding cancer treatment? So, what is,
Okay.
how do you,
Let me give you a simple example. 0.1% of the human genome of 9 billion people are sequenced today. So you have
okay.
9 billion to go.
Mm.
0.1% of the 9 billion are sequenced today. You have 9 billion people in the planet are your customers. So everybody needs to be sequenced. So what everybody will have is their own sequence, is their own human genome, right?
Hmm.
And
Hmm.
we proposed several years back that every baby which is born, we should sequence and have a baby vault. Even that was
Right.
proposed, but we didn't do it.
Okay.
See, today, if you look at Middle East, the consanguineous marriages are plenty.
Yes,
What I mean
absolutely.
by that, the cousins and the relatives, they all
Hmm.
get married actually. So the
Hmm. Hmm.
blood-related disorders are very many. The genetic disorders are very many there. Even in India,
Exactly.
in Tamil Nadu, mama and
Yeah.
that sister, the son,
Yes.
all these people get married actually. So this
Hmm.
is very
Hmm.
well known. So
Hmm.
this is something we need to be very aware of. And another area that I am really concentrating on is rare diseases.
Yes.
So and newborn screening and rare
Hmm.
diseases. This is very important. And government also now thinks like sickle cell anemia sequencing, sickle cell anemia diagnosis is all important. So it is slowly coming on, but the pace at which we are moving in terms of GDP spending on healthcare is
Hmm.
lowest in the world.
That is unfortunate. That is
Yeah,
unfortunate.
we
Yeah.
need about 3% immediately, 3% of GDP immediately in
Correct.
healthcare,
Correct.
and we need equal amount for education, 3% of GDP.
Absolutely. I mean, many things are a little cluttered and needs to be taken care of in
See,
a
if you
better
want
way.
to see 2047, a developed India, like a large Europe,
Correct.
bigger than Europe, let us say, continental Europe. So
Mm.
we need a lot of sacrifice to be made. That sacrifices will have to come from scientists in India, and
Mm.
academicians have to spruce up. The academicians
Yes.
are not coming up with any technology that can be absorbed by industry. Academicians are in their own shell. Why not the academicians come out of their shell and start companies like Americans?
Absolutely.
How is it cozy job to you in sitting in a government job? Why don't you come out? That is the question.
Correct, sir. Correct.
And a lot of
So,
professors, like for example, there was a company called Ambion. He was a
mm-hmm.
former professor of the University of Texas. The question
Mm.
is, I don't know, just to blame the academicians, there is no ecosystem also. Even if
Yes,
some
that
professors want to come out, they are not supported,
they are not supported.
and
True.
they don't get funding. See, if there is enough support, enough ecosystem, they would have come out. So it is both
Absolutely.
ways we are getting affected.
Correct.
So the
Correct.
biotechnology industry itself is in the hands and clutches of few people who are either top business people and small companies, like 99% of small companies which are just 5-man show or 10-man show. So this is the problem. So one or two companies in biotech is not enough. We need tons of companies which are at least 5,000 crore turnover then to make an industry. There is no
Correct.
critical mass.
Mm. Mm-hmm. So,
Startups are good to do innovation, but
correct.
the big companies help the small companies to innovate.
Mm.
That is the ecosystem in the US. If you see the merger and acquisition in the US in genomics, always somebody is buying out somebody else.
Yes.
It is not that Pfizer does everything. It is not that GSK does everything. Even
Mm.
GSK and Merck and Pfizer, they collaborate.
Correct, sir.
So there is an active collaboration between the multinationals. There is an active collaboration between multinationals and small companies. And the professors have early stage technologies. If you look at MD
Mm.
Anderson, University
Yes.
of Texas system. You have all the early stage technologies, so you can walk in and pick the technology that you want. You can develop. You can start companies. I can cite you one company from University of Texas in the early 90s and mid 90s called Tanox Biosystems. A Chinese
Yes.
entrepreneur. They sold their company to Genentech. It is a monoclonal antibody company. So you are talking
Correct.
about 94 monoclonal antibody was very early. At the time, only replacement therapy was there.
Mm.
So replacement therapy in the sense, okay, insulin type of molecules. I have a particular protein that is required. I'll give it to you. It is like a CNTF or NT345, NT5, several molecules that I worked on. Like all these
Yeah.
molecules. At the time, only replacement therapy. So monoclonal antibodies to come to India, it almost took 2010, 2015. So one of my
Right.
presidents in the company was the first person in Missouri to start the monoclonal antibody plant. And several
Okay.
companies in the US at the time, 92, 93, were planning for kilo scale up lab. And 91, we didn't even start. We didn't start actually.
Right.
We were
There's
just
lot
before.
of lot of path or gap that we really need to,
So how to
fast
fill up the
track,
gap,
catch up.
Now the
Yes.
time to allow this small subfields like genomics, proteomics, metabolomics,
Correct.
synthetic biology, you allow it to grow. So these are all
Yes.
subfields of biology. So
True, true. So, sir, I think it's very interesting talking to you, and almost we're at the concluding part of a conversation. But I think it's so interesting that we can go on and on. So, like, sir, how can I mean, this would be my last question, and maybe I think you can enlighten us very well in this. That because being a healthcare professional myself, this is something which is very interesting for me. Like, how can genomic data basically can be used to predict a patient's response to cancer treatment, because we're talking so
cancer,
much about cancer and cancer therapies, so much is happening, but even I always think that we don't have a kind of very handy solution to fight this. So is would genomics give us that power one day, where we have a real solution? So I would really like to know your valuable insights in this.
I think the only way forward is a genome guided therapy. And
Correct.
the way we can assess a particular patient, whether she is
Hmm.
responding or not,
Hmm.
upon therapeutic intervention with a particular monoclonal antibody you're talking about, or a small molecule you're talking about,
Okay.
their survival rates and remissions, and looking at all these factors.
Hmm.
And I have seen, like, for example, this Vas Narasimhan, who is the president of CEO of Novartis, mentioned that their CAR T therapy is really successful.
Hmm.
And
Hmm.
not only that, they are also working on radioligand therapy. So
Yes.
this is a known fact. And therefore, as far as breast cancer, let us say, and you have a number of drugs that are available for breast cancer, I
Hmm.
think reasonably we have two different criteria to screen the patients, to do liquid
Okay.
biopsy in patients and tissue biopsy in patients.
Hmm.
Then we can whole genome sequence, not whole genome, let me say whole clinical exome or this kind of stuff. And they're looking
Hmm.
at actionable mutations, then looking at a particular drug in therapeutic intervention. To
Hmm.
come to your point, how do you tailor make a particular drug is based upon genome data, analytics data,
Hmm.
and how you are able to assess a particular patient is responding or not. It is something clinician can tell. Since I am not a
Yes.
clinician, how they are assessing this, and definitely these drugs are proving to be valuable. I have no doubt in my mind. But
Hmm.
are they completely curing? Is a question that I have for clinicians also.
Hmm. Hmm. All right. So I think, sir, my last question to you would be like, what would be your message to the budding, aspiring people who want to step or make their career,
I
bring
think
themselves into the field of biotech or interdisciplinary? I think this is something which is lacking at our, at least India. I think interdisciplinary science, in the way sciences should be understood, as you mentioned in the beginning regarding the education system and stuff. So I would really like you to, give your valuable suggestions to the young minds, so that at least they get a path of thinking and they can actually be the forerunners in the future to change in a positive way.
I think there are a lot of good students. They are
Correct.
misdirected. Lot of good students,
Yes.
a lot of talent in India. Talent
Correct.
is, we are the suppliers for talent to the whole world. I can tell you.
Yes.
If I can go abroad, come back to India and do all these things. Having been from PhD College of Arts and Science, I know my junior Dr. Raja Mogasi Mangalam from PhD College of Arts and Science. He is an entrepreneur, genotypic technologist. I know my senior Dr. P.M. Murali from Able President. He is from PhD College of Arts and Science. I know Dr. Ramachandran Srinivasan is J Oil CEO in Singapore, PhD College of Arts and Science. We produce a lot of good students from a small college,
Correct.
which had only theory, no practical ability. So it doesn't take anybody to develop themselves, even if they are from a sleepy town.
Correct.
The point is, if you are ambitious, if you are willing to look at science, if you are willing to look at science as a career, and not any other greener pastures like IT or any other, you are welcome to take that. I'm not saying. The question is, if you want to be a born scientist and born science degree holders and your other graduate education and then followed by training and all that. Early promise, let me remember, I told you. Dr. Hegeman told, early promise. He is from a Nobel Prize winner student, PhD student. He has written a book in ACS book. He said, those students who get in very early in science will succeed. That is the theme here.
Right, sir. Right. Thank you so much, sir, for this.
You are very, very, very interesting anchor, and you understand science. That is more important. And I was even asked by many people, general press people, but you have been very inquisitive. I like the way you have interviewed me. Thanks a lot.
Thank you, sir. That means a lot to me. That appreciation from you means a lot to me. So thank you so much, sir, for being on my show, and it was extremely interesting. And thank you for giving so much time in spite of your busy schedule. I'm very delighted to have you once again here, and I think in future we will come up with more talk shows like this, where we can where you can really highlight the real issues, or you can actually provide a pathway to the young minds, as you rightly said. That so like as I mentioned, like when we were communicating, like I also run a e-magazine which is through which I talk about interdisciplinary understand science in its interdisciplinary way. So it's a completely free e-magazine. So I'll be very delighted to have your contributions there also, because there's no charges. We don't put any that restriction, because I think knowledge and science at some point, apart from business, we also need to work on these areas so that it has a free flow.
It must be an open atmosphere, open talk, and open exchange of ideas.
Absolutely.
Science is borderless, boundaryless. It is global.
Correct, sir. Correct. Thank you so much, and thank you once again for being here. Thank you, sir.
I really appreciate your interview. Thanks a lot. Thank you.
Thank you.